to Medical Dialogues. I'm Roshni Dhal. Let's look at today's top health highlights. Researchers provide insights on how to overcome resistance in cancer therapy. The latest developmental drugs, particularly for the use in oncology, rely on the targeted degradation of harmful pathogenic proteins. In a recent study, researchers at CeMM, the Research Center for Molecular Medicine of the Austrian Academy of Sciences, and the University of Dundee in UK, identify potential resistance mechanisms and provide insights on how to overcome them. Traditional targeted cancer therapies mainly rely on drugs that bind pathogenic proteins and inhibited their function. The latest development of drugs has brought forward chemical molecules known as degraders, which force the targeted degradation of disease-relevant proteins. This method of targeted protein degradation is not only more efficient but also better at overcoming potential drug resistance. In this study, the authors identified a variety of mutations on E3 ligases which mediate resistance in cell cultures. These include mutations already identified in patients whose therapy with degraders were unsuccessful. George Winter, who supervised the study, explains that we tested a large number of different proteins and degraders. On one hand, we see that some mutations in E3 ligases prevent defective protein from being recruited. At the same time, however, we see that some of these mutations exhibited high sensitivity to chemically modified degraders. Therefore, mutations can still be targeted by changing the chemical structure of the degrader. In other words, resistance can be overcome. This demonstrates that the results of this study can have clinical relevance as well. Traditional therapeutics that take the approach of blocking an isolated function of a protein only reach about 20% of our proteins. For the other 80%, there are no suitable binding sites, and as a result, they cannot be targeted. We see great potential in this new generation of drugs using targeted protein degradation to significantly increase the space of targetable proteins and potentially reach the remaining 80% of proteins. The authors concluded that the study results makes an important contribution to the improved design of degraders. Cancer patients in particular could benefit from a clinical application of the findings in the future. That's all for today. Stay safe.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.